So we're going to have a 10-minute presentations with 5 minutes for Q&A in between. So I'll go ahead and get started. Excited to present our first presenter, Dr. Erin Bivins from the University of Notre Dame. Go ahead and share your slides, Erin.
Okay, I think they're shared now. Hi, everyone. My name is Erin Bivins. I'm a postdoc at the University of Notre Dame. And the title of my talk today is Microbial Water Quality in Intermittent versus Continuous Water Supply in Nagpur, India. This is a project I led as a PhD student at Georgia Tech with Joe Brown. And just go ahead and acknowledge the funding sources, the EPA, and then I was a Fulbright Nehru Scholar in India, which was a fantastic experience. The AWWA, as a Fulbright Nehru, I worked at CSIR Neri with Dr. Pawan Labhasidwar, who heads the Water Technology and Management Division there. I worked alongside Sonal Wankade and Rajashree Hajare. And then, of course, Orange City Water, who is the operator of the water supply there in Nagpur. So a bit about intermittent water supplies. An intermittent supply is a pipe supply that is consistently interrupted, usually as a strategy to manage scarcity, either of water itself or of the inputs that you need to produce water, such as skilled labor or electricity, things of that nature. And because these supplies are interrupted, the multiple barriers that are used to keep them free of contamination are interrupted as well. And so they're associated with degraded water quality and subsequently endemic and epidemic diarrheal disease. And so in a previous work, we estimated that around 925 million people are exposed to IWS globally. I think this is probably a lower bound. I think it could easily be more. And among these 925 million people, we estimated that there could plausibly be about 17.2 million waterborne infections annually. So when we talk about intermittent water supply in India, it's important to note that as of now, no major city in India offers continuous water supply to its population. So that's about 460 million urban dwellers that are exposed to IWS in India. Nagpur, however, is poised to become the first major city in India to provide its populace with continuous water supply. And that process began in 2011 when the Nagpur Municipal Corporation contracted with Orange City Water to transition the supply to 24/7. And so from 2011 to 2016, approximately 500 kilometers of pipeline were replaced, meters were installed, and CWS has actually been initiated in 10 of 64 command areas. I think now, in 2017, that was up to 13, I believe, which a command area is just a hydraulic zone in the system. So we leveraged this transition to study microbial water quality in intermittent versus continuous areas. And our sampling protocol consisted of, we sampled at taps as well as elevated storage reservoirs. We used traditional grab sampling. So we would collect around 500 milliliters. But then also we use these dead-end ultrafilters as shown here, and we would filter anywhere from 20 up to 120 liters of drinking water and concentrate that into about 4 milliliters. And so we used some culture-based techniques for things like thermotolerant coliforms and E. coli, but we also used molecular methods, droplet digital PCR, to assay the DEUF concentrate for a suite of genes associated with various waterborne pathogens. As I mentioned, the water supply at this time was under a transition, and so we sampled in one continuous water supply area, Lakshmi Nagar Old. And this area received its drinking water from the Pinch 2 water treatment plant. From there, it's pumped to the elevated storage reservoir, and from this elevated storage reservoir, it's distributed continuously to household taps, as shown by the blue arrow. Conversely, this treatment plant also supplies water to a couple of intermittent water supply zones, Reshamba, Karve Nagar, and Kamla. The water is pumped to the ESR, but from the ESR, it is distributed intermittently within each command area. And so we hypothesized that this intermittence would be associated with a degradation in water quality, as portrayed here with the brown water in the sample from the household tap. And so we have a lot of data. I'm going to hit the highlights. First is the culturable E. coli prevalence. This is in 100 ml grab samples, which we processed using membrane filtration. Among the 64 grab samples from household taps, 28 of those were positive for culturable E. coli, whereas in the continuous water supply zone, out of 24 samples, only a single sample was positive for culturable E. coli. And so we do see a significant increase in the prevalence of culturable E. coli at household taps served by intermittent water supply. The figure on the right here shows the E. coli counts as observed in IWS versus continuous water supply. But of course, because we only had one countable plate in the continuous water supply, hypothesis test isn't possible there. When we look at the gene detections of genetic targets associated with pathogens, we see consistent detection across all targets in samples from household taps served by IWS. And when we aggregate those detections, we see that 61% of the samples from household taps receiving intermittent supply were positive for a gene target associated with a pathogen, whereas only one sample was positive from the continuous water supply zone. So when we look at the uncertainty around these intervals, we see that intermittent water supply is associated with an increased prevalence of genetic targets associated with pathogens at the household taps compared to continuous water supply. Another important theme that seems to be emerging is the heterogeneity of microbial water quality within and between intermittent water supplies. So not all intermittent supplies are equal. For instance, in Nagpur, as shown on the right, we see the proportion of grab samples positive for E. coli ranged from 80% in one area to 28% in another. And this is consistent with a previous study in Hubli Dharwad, where the E. coli proportion positive ranged from 4% to 80% when stratified by hydraulic zone. And then there's another study conducted in Panama where actually E. coli was detected in only one of 423 grab samples. So again, we see this context-specific variables probably are in play here when we're looking for contamination and microbial contamination in intermittent water supplies. Some of the limitations of our study, the small sample size makes it very hard to compare, especially when you're working with microbes. Again, 90 dead end ultrafiltration samples and about 150 grab samples. I think it's important to recognize that these dead end ultrafiltration samples are a total volume of almost 7,000 liters. So a tremendous amount of water that was filtered and concentrated. And as an example, in our 9 DEUF samples from continuous water supply taps, we didn't detect any culturable E. coli, and that's about 1,000 liters of water. Of course, these molecular targets are proxies for viable pathogens. And then our sampling was limited to 3 intermittent water supply zones and 1 continuous water supply zone owing to the logistical constraints. So I think there's an important implication for the SDGs, which is that, the safely managed drinking water is defined as located on premise, available when needed, and free from contamination, fecal contamination, in this case, E. coli. And we see that IWS, especially in South Asia, is unlikely to meet these standards. Again, approximately 44% of our grab samples from IWS taps had culturable E. coli in them. And so of these 5.3 billion people currently estimated to have access to this safely managed drinking water, that number includes 925 million served by IWS. So there's, I would say, increasing evidence to indicate that these supplies do not confer the health benefits we'd associate with safely managed drinking water services. I guess at this point, I'll happy to take any questions. Thanks for your time.
Thank you so much, Aaron. So we already have a bunch of questions in the chat. So I'll just remind everyone, we only have 5 minutes for questions, but we do have a messaging feature on the conference platform. So there are two pretty much identical questions, and you can see on the screen that Elizabeth is sharing where you can ask questions. And two questions about why there were more crypto positives in continuous water service than IWS, and why you think that might be.
Yeah, actually, so the percentages there are a little deceiving. There's actually one cryptosporidium detection at an IWS tap and one at a continuous water supply tap, but the percentages look different because the sample size is different. But yeah, as to why that might be, I'm not sure, but that was the case.
Great, thank you. And then there's a question about residual chlorine levels. Were they comparable in IWS and CWS zones?
Yeah, I can't remember off the top of my head. I do think that a lot of our supplies were actually, the residual chlorine was quite low. I think the India standard is 0.2 milligrams per liter for distributed drinking water. And I think about half of our samples didn't meet that criteria in both intermittent and continuous water supply zones.
Great, thank you. Another question about heterogeneity, but also, let's see.
Yeah, the heterogeneity issue is really interesting, and I think it has to do with, you know, the how many potential fecal sources there are in an area, how close those sources are to the water lines, and then of course, you know, how these intermittent supplies actually function hydraulically is quite complicated. And so that could create a lot of variation in the opportunity that contamination has to enter the distribution system. So I think that's a very important point that, you know, in Panama, we didn't see as much E. coli, whereas in South Asia, we seem to detect E. coli about half the time in the grab samples. So that would indicate that, you know, broadly classifying all IWS as equal is probably not the right approach.
Thank you. And it looks like there's a question that says, how did Nagpur transition to CWS from IWS, and how viable or likely is a similar transition in other major urban centers in India?
Yeah, that's a awesome question. You know, Nagpur has had great success with their project so far, but all of these changes are premised upon financial solvency. And so Nagpur has spent a tremendous amount of time reducing their non-revenue water and installing meters and upping their bill collection efficiency. And those are actually the first steps in this transition. Dr. Labhasitwar and his team have actually written an interesting paper that looks at more of the kind of the logistics of how Nagpur has gone about doing this, and that's a great paper worth visiting.
Thank you. And you're getting lots of questions, so that's a good sign. What could be some strategies for low and middle income countries to establish monitoring systems of parasites in drinking water?
Yeah, that's tough. You know, I mean, ultimately these methods that we implemented in India are difficult to achieve. You know, these ultra filters are about $15 each, and I think that's a huge area of opportunity to come up with some cost-effective, some low-cost and sensitive methods for detecting these types of pathogens. And I think that there's some exciting opportunities now that are starting to emerge with things like PCR, and then of course now LAMP has come onto the scene, the loop-mediated isothermal amplification. I think that shows a lot of promise. So that's something I'm interested in carrying forward.
Last chance for questions. We have time for about one more, but you've also gone through a lot of questions in a short amount of time, which
Trying to be efficient.
Thanks. Oh, okay. Are there chlorine injection points along the pipeline from initial treatment to point of use?
Yeah, that's an excellent point. So these elevated storage reservoirs actually are dosed with chlorine. There's some gentleman whose job it is to go up about 40 feet of stairs and dump chlorine powder into these elevated storage reservoirs. And so I guess depending on his mood each day, the water is either chlorinated or not. But supposedly, yeah, there is consistent chlorination at the ESR. The water is allowed to mix for about 30 minutes and then it's distributed, at least in the case of an intermittent zone. But based on our residual chlorine results, it seems like that doesn't always happen.
Great. Well, thank you so much for a great presentation, Aaron. If you could stop, well, you're not sharing your screen, never mind. We're going to move on to our second presenter, who is Andrea Martinsen from the CDC. As a reminder, we have 10 minutes for the presentation, followed by 5 minutes of Q&A. So go ahead, Andrea.
All right, thank you, Nikki. Good afternoon, everyone. As Nikki said, my name is Andrea Martinsen, and today I'll be presenting findings on behalf of our team at CDC, as well as our partners in Zimbabwe on an evaluation we did on inline chlorinators on hand pumps. To begin, just a quick overview of the wash situation in Harare, which is the capital city of Zimbabwe. Although the majority of properties are connected to the piped municipal water supply, many households use alternative groundwater sources as their primary source of drinking water. Aging infrastructure, economic instability, population growth, as well as recent droughts have led to increasing water shortages and aesthetic concerns of the municipal water. In the high density suburbs, one of the most common drinking water sources are the community boreholes installed with the Zimbabwean type B bush pump, which is pictured here on the top right. There have been multiple typhoid and cholera outbreaks in the past decades, particularly in these high density suburbs, and they've been associated with municipal water shortages and increased use of contaminated boreholes and shallow wells. As a response to a large typhoid outbreak originating in one of the high density suburbs in 2017, Oxfam in Zimbabwe began a project to design inline chlorinators to be installed on the bush pumps. And a close-up of the box containing the inline chlorinators is pictured here on the bottom right. Water quality testing showed that some boreholes in this area were contaminated, and inline chlorinators were selected as a point of collection treatment method due to the dependence on bush pumps. CDC began working with UNICEF and the National Institute of Health in the city of Harare and Oxfam in late 2018 during a large cholera outbreak in Harare. And then we conducted this evaluation in mid-2019. And the two main objectives were to document their performance in providing chlorinated water and to assess their use and acceptability. The evaluation was comprised of 5 main components: the performance assessment, a household survey of beneficiaries, interviews with water point committees, focus group discussions, and key informant interviews with stakeholders. For the purposes of this evaluation, I'll just focus on the first 2 components. And first, I will present the methods and results of the performance assessment, and then go into detail on the household survey. And then finally, a recap of conclusions and recommendations. So to evaluate the performance, we assessed 66 inline chlorinators installed on bush pumps in Harare. And several performance criteria were included, with our primary parameter of interest being free chlorine residual, or FCR. First, we tested the FCR from a bucket filled from each chlorinator to see whether the device was functioning and producing chlorinated water, and if it was dosing at the recommended levels. Second, we tested whether the pumping rate affected the chlorine dose. Third, we tested the consistency of dosing while filling up the bucket by testing FCR at different time points during the filling. So first, when first starting to pump, then after 30 seconds of consistent pumping, and then finally after letting go of the hand pump handle. Lastly, to assess consistency of dosing over time, all of the inline chlorinators evaluated during this assessment were monitored weekly for FCR. Of the 66 inline chlorinators visited, FCR was detected from 33%, and 14% provided an FCR at the recommended levels between 0.5 and 1.0 milligrams per liter. Of note, 77% with no detectable FCR had a chlorine sleeve present. which suggests that even when chlorine sleeves were present, the current design may prevent consistent discharge of chlorinated water from the hand pumps. We also found that the pump rate determined the chlorine dose applied, with slower pumping resulting in a lower FCR. And then to better understand the consistency of chlorine dosing during container filling, we conducted a repeated measure analysis using the 3 different samples that I detailed on the previous slide. This model found no statistically significant relationship between the FCR detected at these different stages of pumping. And this is shown in the graph here on the bottom right, where each line depicts a different inline chlorinator. Now moving on to dosing over time. After the initial performance assessment and to prepare for weekly monitoring, we adjusted all the chlorinators to an FCR of 0.5 and replaced chlorine sleeves where necessary. After these adjustments, 88% of the buckets filled from the inline chlorinators had a detectable FCR. City of Harare staff then visited each inline chlorinator weekly for testing. And the FCR went down over time with the proportion after 1 week at 54% and then down to 34% after 2 weeks. Again, a repeated measures model was run, this time comparing the FCR levels after adjustment with the first 3 rounds of monitoring data. And with this, we found a statistically significant relationship in the downward direction over time. Now I'll move into the methods of the household survey we conducted to evaluate use and acceptability. We conducted a 2-stage cluster sampling in 4 high-density suburbs, and the target population was divided into 2 separate groups due to differences between the suburbs. From each group, 15 inline chlorinators were randomly selected, and around each inline chlorinator, 24 households were systematically selected. To do this, we first selected 4 randomly generated points using QGIS within a 200-meter radius of each inline chlorinator, and then drew buffer lines at 30-meter increments around. Then at the intersection between these radii and the buffer lines was where the households were selected for surveys. And this is shown on the map on the top right here. As previous studies had shown in Harare, boreholes were the most commonly reported primary source of drinking water, with 2/3 of households reporting it as their primary source, compared with just 27.6% reporting municipal water. We also found that residents reported to trust borehole water more than municipal water, with 89.2% reporting it to be safe to drink, compared with just 53.3% for municipal water. To assess use, we asked several different questions on past and current use. Self-reported use varied from approximately 80% of households reporting to have ever used an inline chlorinator. This went down to about 65% who reported to use in the past week, and about half with stored water at the time of the survey from an inline chlorinator. Reported use in the past week decreased at farther distances from the inline chlorinator, with over 80% at the 30-meter mark, and then 60% at 210 meters. We compared households at 90 meters or less with those at more than 90 meters, and the closer households had a 1.8 times greater odds of reporting to use the inline chlorinators. When asked what their preferred method of treatment was, 61.9% of households preferred point of collection treatment with the inline chlorinators, compared with 30% of household water treatment. And some focus group discussion participants cited that they perceived the inline chlorinators guaranteed safe water for all, that it was more affordable and reliable, and they also didn't have to worry about incorrect dosing as they did with household water treatment. However, contrary to this perception, only about 1/4 of households had reported to receive messaging about the inline chlorinators, including when it's working, when there's chlorine present, and when there's adequate dosing. So in conclusion, 1/3 of the inline chlorinators had a detectable FCR during the initial assessment. And furthermore, dosing varied during pumping and by the pumping rate. Additionally, the proportion of inline chlorinators producing chlorinated water went down over time during the weekly monitoring. And contrary to the previous monitoring reports that suggested that this may be due to lack of community management and inadequate timely replacement of chlorine sleeves, we found that many of the non-functioning chlorinators actually still had chlorine present. So our assessment showed that they needed much more frequent monitoring and adjustment as what was previously thought. In terms of use and acceptability, the inline chlorinators were well received in the high density suburbs and may be an alternative method of water treatment during cholera outbreaks or other waterborne disease outbreaks. However, while inline chlorinators were preferred and potentially more trusted than household water treatment, most households report not receiving information about the FCR levels, and there's a disconnect between the trust and the actual functionality of the inline chlorinators. We recommended potential design modifications to the implementing partners, such as changing the size and quality of the component parts and to conduct additional pilot testing. And then to address this disconnect between use and trust and lack of information, we recommended that more community engagement is needed, and a monitoring system of FCR should be set up to allow for more frequent adjustments and to share results on a routine basis with beneficiaries. And finally, I'd like to thank our many partners in Zimbabwe, as well as our donors that helped to make this evaluation possible, and special thanks to these individuals. And at this time, I'd like to open it up for questions and also introduce my co-investigator and colleague, Rajasingham, also from CDC.
Thanks so much, Andrea. Really appreciate you sharing your presentation. We have a question here. As you can see, it says, how long is chlorinated water typically stored and used in people's homes in this setting? And did the research also look at FCR levels in the household point of consumption?
Yeah, so to answer the first question, I think that because of the prevalence of boreholes, it wasn't stored for that long because there was available water most days. We did also test FCR at the household, and I didn't present those results in the interest of time, but did find that about 50% of the households that had reported to collect from the inline chlorinator, and that there was a detectable FCR at that inline chlorinator, had a detectable FCR in the household. So 50% had an FCR, 50% did not. We need to do some additional analysis to also look at time of collection, but that's to be done in the future.
Oops, I was muted. Thank you. There's a question here that says, do you think there are designs for inline chlorinators that may be more resistant to this impact happening?
Yeah, and I don't know if you can unmute yourself. I mean, we also looked a little bit on the submersible pumps that had a continuous flow. I think it's much easier to control the dosing on this type of diffusion erosion tablet inline chlorinator. Anu, do you have any additional comments for this question?
Yeah, so what we found was the consistent flow inline chlorinators had appropriate dosing. It was because of the variability in how you pumped and the volume of water that was actually passing through the chlorinator that we saw this inconsistent dosing.
We have time for maybe 1.5 questions, we'll see. But here we have a question from Sarah Young that says, I'm curious to know more about the technology and product itself. Is it available on the market there? Is there any specifics? Are there any specifics about it that we can look up? What adjustments were made?
Yeah, so it's locally produced. So it's available on the market in Zimbabwe, but not outside of Zimbabwe, and specific to the bush pump, which is common in the country. Anu, do you want to talk a little bit more about the design modifications we recommended?
Sure, because we found that the volume of water that passed through the chlorinator basically determined the dose that was applied. We suggested and recommended that they increase the size of the tubing that led from the inlet hose into the chlorinator and out of the chlorinator, as well as there was some design modification to the size of the hole that led the water in and out of the chlorinator. So with those design changes, they could potentially have more water passing through and increase their dose, as adjusting became the trickiest part of this chlorinator, and making sure that over time there was chlorine.
Great, thank you. Unfortunately, we have a couple remaining questions, but there is a message feature on the platform, as I said, and the chat will remain there. So speakers, if you want to check out the questions that I wasn't able to relay to you, that'd be great. We're going to move on to our third and final speaker. This is Anna Murray from Charity Water. Anna, could you go ahead and share your screen? And go ahead.
Thanks so much. All right, so thanks everyone for joining this presentation. My name is Anna, and I'm the Senior M&E Manager at Charity Water. And I'm presenting this work on behalf of Justine Rayner. So she carried out the evaluation and did most of this work. So if there are any questions at the end, I'll do my best to answer them as best I can. So getting into it, a biosand filter is a household level, intermittently operated slow sand filter. So it's comprised of a casing, which is usually concrete or plastic, and then there's sand and gravel inside the filter. So water goes into the top, down through the sand inside, and then out an outlet into a storage container. So the filters remove pathogens that cause diarrheal disease through mechanical trapping, as well as natural die-off. And there's a layer, a bio layer that forms at the top of the filter that helps as well. These have been evaluated in many settings and have been shown to reduce diarrheal disease as well. I also want to introduce a larger scale filter, which has been scaled up for use in schools. And so this type of filter was developed jointly by Samaritan's Purse and Clear Cambodia back in 2011. So it uses similar principles to the household scale biosand filter. So in here, the top filter holds the water to be treated. The sand is in this middle tank, and then the stored water is in the largest tank there. And so Clear Cambodia, they're an NGO based in Phnom Penh, and they implement a very large scale biosand filter program. The program has been funded by Charity Water directly and indirectly since 2009. So in that time, Clear has installed over 240,000 household biosand filters, as well as about 800 of those school biosand filters. And so their household program is community-based and it's demand-driven. The households build their own filters, and then the staff helps with the installation and follows up with the households 4 times within the first 2 years. In the school program, it often includes latrines and hand washing stations and wash education as well. So the household portion of the Clear Cambodia filter program was evaluated back in 2010. And some of the results are shown here on this slide. And so at Charity Water, as a major funder of this program, we were interested to commission a similar follow-up evaluation almost a decade later to see how things were going later. So the core research questions we're looking at were from the household filters. How long do people continue using the filters, and what factors are associated with that sustained use? And then what is the effectiveness of filters at removing E. coli? And then in addition to that, we wanted to look at the school filters and how effective they were at removing E. coli. So in order to answer these questions, we did a cross-sectional evaluation in 2 provinces. So the provinces were selected because they were both included in that 2010 evaluation. And also they were ones in which the filter distributions had gone back many years, as far as 2009. And these 2 provinces also had school filters available. And so within each province, 7 villages were randomly selected, and then households were randomly selected in each of those villages. The schools were selected by convenience sampling close by to the area that the randomly selected villages were. So in total, The sample included about 300 households and about 50 schools. So at both the households and the schools, we had surveys conducted, and the biosand filters were observed for a variety of things. And then water was tested as well. So there were 3 samples taken for the E. coli testing. The first was untreated water. The second was treated water that was taken directly from the filter outlet. So like is shown in this photo here. And then the third sample was taken from the storage container and usually served in a cup as the person would drink the water. And water sampling was done at about half of the households and all of the schools that were sampled. All right, so I'll start with the results from the household filters. Overall, we found that 71% of the households were still using their filters. And looking at that by the 2 provinces, we actually saw a lot of variability. So 81% of households in Sfaerang were using their filters, but only 56% in Kampong Thom. So some possible explanations for this. Kampong Thom seemed to have a higher socioeconomic status. There's more use of bottled water and also more availability of other household water treatment options. And I'll discuss a few other things later on about the use. So the first research question was about the time in use. So in this evaluation, households had been using the filters for an average of 6.5 years. And I want to note here that about a quarter of the households had been using the filters in excess of 8 years, and some up to 14 years. So we think this may be among the longest documented household biosand filter use. And then interestingly, even among those who had no longer been using the filter, we did look at how long they used it before they stopped. And that was on average about 5.2 years of use before they stopped. And so now looking at what factors were associated with the sustained use. We performed a logistic regression. So the outcome variable was the filter being in use. We also controlled for the province and the age of the filter. So some of the significant variables are listed here. I want to highlight a couple things. First, the taste seems to be an important driver in the adoption of the biosand filter. And also, I want you to look at this odds ratio with the deep tube well water source. This was a little bit unexpected for that to be such I guess, high odds ratio. And this is also something that came up in that 2010 study. We think it might also be related to the taste, as water from some of these deeper root tube wells may have a metallic taste that the filter could be improving. So moving on to the water quality. This graph is showing the E. coli results categorized by the diarrheal disease risk level. So that's represented by the different colors here. On the left, you see the untreated samples. The middle has the samples taken directly from the filter. And then on the right is the samples that were treated and stored. So I want to focus on just the low risk level for diarrheal disease. So if we look at this level, we see almost half of the untreated samples complied with that. And then they increased to 80% of the samples that were treated as they came out of the filter directly. But then that dropped back down a little bit to 67%. So we see the filter is improving the quality of the water, but that the water people are actually drinking, about 1/3 of those samples still had medium to high risk of diarrheal disease. So the filters are working, but they're not working perfectly in all cases. And looking at this a different way, if we want to compare the untreated water to the water taken directly from the filter outlet, we saw an 88% reduction in E. coli. And comparing the untreated water to the stored drinking water, that was a 70% E. coli reduction. And so I just want to note here too that the influent water concentration was fairly low. So we had 14.7 CFU per 100 milliliter. And so as a result, it's not possible to demonstrate a very high percent reduction because the water starting out was not so contaminated on average. Okay, so moving on to the school filters. We found 90% of the school filters were in use, and that was after about 5 years following the implementation. The respondents, which were principals and teachers, they reported seeing less absenteeism and also increased student participation after the filters had been installed. We do not have any observational data to back that up. That was sort of anecdotal from the respondents, but that is positive to see. And the E. coli results for the school filters are shown here. Again, I'll just highlight that low risk level. So here we saw the influent water, the untreated water, was starting out actually pretty good. So about 80% were in that low risk level. And that improved to nearly all of the samples that were treated by the filter, and then decreased a little bit in the stored. So again, looking at the percent reductions, we see 75% E. coli reduction directly from the filter, and 60% reduction when we compare the untreated to the stored water. And again, here we saw even lower E. coli levels at the influence water. And so it was only on average 2 CFU per 100 mil. And so this may not represent the full potential of the filters because it wasn't really challenged in this field setting. So in summary, filters were used by 71% of households and 90% of schools up to 14 years after the program implementation. Even households that stopped using the filters had used them almost for a little over 5 years. The filters were effective at removing E. coli. We think this may be the first independent evaluation of this larger scale school biosand filter, so showing that it's a promising technology. And again, E. coli recontamination is an issue with biosand filters. It's been a persistent problem in many similar evaluations. So I'll leave you with a couple of recommendations. It would be good to validate these findings of the school biosand filter, preferably in a situation where they're challenged a little bit more with more contaminated water. Second, we really need to emphasize safe handling and or residual disinfection to prevent recontamination of the stored water. It's a persistent problem. We have to figure out how to best address this. And then finally, one of the major benefits of the biosand filter is its long lifespan. So we saw that here, and this underscores the need to sustain supply chains. So things like additional sand, so that users can keep using the filters over time. And I just want to acknowledge the implementing organization, Clear Cambodia, the survey team, all the respondents, and Charity Water funded the study here. And I'm happy to take any questions.
Thank you, Anna, for your presentation. There's already a bunch of questions, but unfortunately we will only have time for a couple of them. So just take a look at the chat afterwards.
Yeah,
so,
I'm happy to follow up with more in writing.
First, there's what was the filter distribution process and what households and schools received the filters?
The filter distribution process, this is challenging my knowledge of the program a little bit. So I believe that Clear, so it's demand driven. So they work with community leaders and then individual households. I think when they hit a critical mass of people in a particular village that want the filter, then they will run a training session with the household members in that community who want the filter. So it is demand driven. There's also a small contribution. So it's subsidized, but the households also have to contribute. And then there's training that's involved when they construct their filters as well.
Great. Thank you. And then a question about the methods. Why was in use defined as being in the past 7 days? Does the biolayer need more frequent use to be effective?
So good question. So the biolayer does need semi-continuous use. So you don't want to leave it out of use for around, I don't know, for like a couple weeks at a time. And so I think 7 days was chosen as more from we think that they're still using it on a regular basis. less and less about the biolayer. If that makes sense.
Definitely. Okay, we'll do one more. Is any consideration made for chemical issues that cannot be treated by the Biosand filter? For example, arsenic that may exceed drinking water standards.
So in this program, they're focusing mainly on the microbial contamination of the water. We actually, as part of this study, we did a small sample of arsenic samples as well to see if the Biosand filter was also helpful in removing arsenic from the water. We did not see many. First of all, I didn't present it here because we didn't have that many samples to really draw a firm conclusion about that. And also there were not many samples that were very high. And so the program is mainly not working in areas that have high arsenic contamination. And so, yeah, there is a bit of literature out there on the Biosand filter and its ability to remove arsenic or other physical chemical components, but we didn't focus so much on that.
Okay, well, thank you, Anna, and thank you to all three of our speakers today. That was great. I definitely learned a lot, and there were so many questions, which means it was a really engaged audience, and you guys did a great job. So thank you. So we're done for this session. So the next one is in 10 minutes. And also just one other announcement, we do have a poster happy hour session today. So please join by, bring a beverage, bring snacks. We actually have a featured wash cocktail that's going to be coming out in an email, so look out for that as well. But thank you for joining us today, and looking forward to seeing you sometime during the rest of the conference. Thank you.